The overall goal of this procedure is to isolate a specific genomic region retaining molecular interactions for identification of molecules interacting with the genomic region.
This method can help answer questions in the genome functions fields, such as transcription and epigenetic regulation. The main advantage of this technique is that it enables direct identification of molecules interacting with a specific genomic region. Demonstrating the procedure will be Toshitsugu Fujita, an assistant professor from my laboratory.
Following the text protocol, engineer the DNA-binding molecules of interest. This entails generating the G-block sequence of interest and inserting it into a retrovirus, for example, to produce the guide RNA complexed with 3-flag-dCas9. Alternatively, 3-flag-tall protein recognizing the target sequence can be generated. Then, following the text protocol, infect the cells with the prepared retrovirus. Confirm the expression of the engineered DNA-binding molecules, and establish the cell line. Now, proceed by cross-linking the cells with formaldehyde. Suspend 20 million transfected cells in 30 milliliters of regular culture medium, and add 810 microliters of 37% formaldehyde for a final concentration of 1%. Incubate the mixture for 5 minutes at 37 degrees Celsius. After 5 minutes, kill the cross-linking reaction by adding 3.1 milliliters of 1.25 molar glycine. Let the cells sit at room temperature for 10 minutes before proceeding. Next,